My name is Jesse Rogers, an ORISE postdoc at US EPA's Office of Research and Development, and I'll be presenting our group's work in integrating high-throughput transcriptomics into a tiered framework for chemical toxicity testing. One of the long-standing issues for chemical toxicity testing is that there are far more chemicals commercially in use than resources to test for human safety. Current testing guidelines require animal studies that cost time, money, and may not fully approximate human biology. And human cell-based assays such as transcriptomics and targeted screening are gaining more attention in the regulatory space. In order to further adoption of in vitro assays, a computational framework is required that can both integrate assay data to build confidence in overall toxicity and link chemicals to key hazards. US EPA is currently developing this framework using high-throughput transcriptomics for initial screening, followed by targeted assays for validation. And our work here demonstrates a proof-of-concept framework linking these data streams and applies it to current chemical screening data. In order to link transcriptomic data to key mechanisms of action, we first identified reference chemicals using RefChemDB, a database of literature associations between chemicals and molecular targets. We applied a procedure including filtration based on literature support, dimensionality reduction of molecular targets, and assignment of individual chemicals to latent clusters, ultimately linking chemicals to putative mechanisms of action based on overall levels of literature support. After cross-referencing the resulting assignments with our current transcriptomic screening data, we found 13 putative mechanisms of action that were sufficiently represented in our data, encompassing xenobiotic metabolism, endocrine regulation, and several other functions. From here, we used previous high-throughput transcriptomics data generated by US EPA, in which over 1,200 chemicals were screened in a multi-concentration format using the TempoSeq platform. Our published pipeline was used for transcript alignment, counting, QC, and differential expression analysis. And we used the TCPL-FIT2R package to estimate benchmark doses at which a chemical causes a change in gene expression beyond a noise distribution. This becomes an estimate of chemical potency for each gene and forms the basis for chemical comparisons. Using the reference chemicals we assigned earlier with benchmark doses for each gene, we generated custom signatures that were uniquely potent for individual classes of reference chemicals. For each gene, we used a one-way ANOVA and Tukey's post-hoc to test for significant differences in benchmark dose as a function of reference class. If a gene was uniquely potent for a single class versus several other classes, then it was assigned to that class as part of a reference signature. This process resulted in 5 signatures representing 4 distinct mechanisms of action. For each heat map shown here, we compared the potencies of genes identified for reference signatures, shown as columns, across individual reference chemicals as rows. Chemicals annotated for each MOA show a distinct potency on a gene level for the matching signature, as well as limited potency or no response for genes and other signatures. We also observed that different signatures were identified between HEPA-RG and U2OS cell lines, suggesting that multiple cell lines may be necessary for assessing a broad range of MOAs. We then generated signature-level potency estimates for each reference chemical using the CompTox HTTr pathway package, which is available on the US EPA GitHub. The package first used a single sample gene set enrichment analysis to estimate enrichment scores for each signature, and then estimates benchmark doses from those enrichment scores. We compared signature benchmark doses on the x-axis to scaled efficacy values on the y-axis for all reference chemicals, and we can see that chemicals annotated for that signature in blue generally have larger effects at lower doses compared to chemicals annotated for other signatures. And this suggests that these signatures can distinguish chemicals by their annotated MOA. We next developed a proof-of-concept framework for toxicity testing by integrating our reference signature analysis with high-throughput screening data of nuclear receptor activity from US EPA's ToxCast program. We intended to identify chemicals that selectively acted on each MOA associated with our signatures, and so we conducted this assessment in 4 steps. We first identified chemicals active for any of our reference signatures, and followed up by comparing signature potency values to estimates of non-selective transcriptional activity. Passing chemicals were then validated using nuclear receptor activity screens in a similar manner, first for bioactivity in orthogonal endpoints, and then for selectivity to those endpoints compared to estimates of non-selective activity. Of the over 1,200 chemicals initially screened with high-throughput transcriptomics, 67 candidates were ultimately validated as selective modulators for one of 4 MOAs using this framework. These 67 validated candidates include marketed drugs for the associated target, such as glucocorticoid receptor agonists, as well as those with known toxic effects, such as aryl hydrocarbon receptor-mediated carcinogenicity by polycyclic aromatic hydrocarbons. And these candidates are further explored in our poster. Lastly, chemicals positive for each transcriptomic signature were significantly more likely to be positive for at least one validation assay, suggesting that such signatures can effectively prioritize chemicals with hazardous potential for further testing. Our study results suggest that integrating high-throughput transcriptomics with targeted validation assays can identify hazardous chemicals and link them to key modes of action. We hope to use such strategies to prioritize data-poor chemicals for further testing, and we hope that integration of additional data streams, such as high-content imaging, will further regulatory adoption of these technologies for chemical toxicity testing. Thank you so much for listening to this flash talk, and please reach out with any questions.